The Cyto Conference organized by ISEC is one of the biggest flow cytometry conference. So we can learn from each other, and then also there's a lot of state-of-the-art technologies and cutting-edge data is presented by the researchers. That will definitely help us develop new assays for the need for the clinical trials.
I think it's quite remarkable that all the inventors of the softwares, the tools, the instruments that we are using on a daily basis, they're just here. So it's great to connect with them. And it's also good, or we value also good relationship with the vendors we are working with. Not only to have them motivated to be ASAP in our laboratory if we have a technical issue, but also just to have cooperations with them on the development of new innovative tools. An example is like our cooperation we have with Slingshot on the development of new QC material for clinical testing of flow cytometry.
Flow cytometry can be especially useful in clinical research because it allows the ability to immunophenotype certain cell subsets as well as assess functionality of those cells. As far as advantages, in the clinical space, there's the ability to monitor disease progression by flow cytometry, just evaluate the immune system in general as well.
One of our posters is about the description of performance qualification of our Cyto instruments. This process is important because it allows to define that the instrument that we're going to use can be used for its intended purpose and consistently over time. And for the kind of testing that we perform, that's the most important thing because we test samples of clinical trials, so we have to make sure that our instruments performs perfectly. And beside the workflow, we also describe some troubleshooting. And we thought that this was very important for the community because not only shows that at Cerba we have expertise to really understand when something goes wrong in an instrument and how we're able to fix those problems, but we also thought that as scientists it was very important to share this with the community so that they could learn and not get into the same mistakes, and also open up conversations on exchange of information so that we could all grow and do better.
We are presenting a poster about our standardization work on a BD lyric instrument. And in that poster we show how we tested the standardization of the instruments over time. So we do show that the instrument is very stable over time, the performance is very well. We also go one step further in QC testing of that instrument by checking the alignment between instruments, monitoring the MFI. And these data do show us that there are differences to be seen between instruments, and these are to be monitored, and it's impacting the selection of the instrument we choose per assay.
What we have done in the past couple of years is to expand, upscale the labs. So upscaling means we invested a lot of new instrumentation, automation as much as possible. So that provides us a better way to upscale the clinical trial samples globally. So one of the posters will explain how we upscale the lab and how we are implementing new processes for antibody cocktailing, data analysis, and then review and release.
I firmly believe that Cyto is the instrument of the future because the clinical trials, and it's just getting, I think, thirstier and thirstier when it comes to gathering information. An instrument like Cyto allows for that because it gives you the possibility to deep characterizing your patient sample and collecting a huge amount of information. We can discover new biomarkers that we wouldn't be able to discover before. The quality of the data is really impressive. We did do some testing with other instruments, running same assays side by side, and there is a resolution capability that is really different and beautiful when you look at your data, and it's completely unexpected. The other aspect is that we can do retrospective analysis with that instrument. So, for example, in cases where big pharmas or sponsors do not yet what to look for, they can, for example, freeze their samples. We run a certain kind of assay, and then they would be able to come back to that sample later and ask to design like a bigger panel and add more markers to it. They can go and learn from the results that they get one day and then go back and say, oh, but let's do that. Or even just save the sample and and reuse it even like, for another trial or something, and still like add all of these markers. So there's really like a lot of potential.
As the technology continues to improve and we learn even more about it, I think being able to use flow cytometry to really interrogate the immune system is critical. There's so much that we don't know about that, and being able to evaluate biomarkers at a very high level, meaning that you can throw a lot at a small volume sample and be able to tell so much about that sample and that person and the disease, as well as any drug treatments that are being offered to that patient, I think is just really critical and exciting in this industry. So looking forward, I think that, as this technology progresses, there's so much that we can learn about ourselves and how to, better help the patient, which is ultimately the goal for all of us.
I would say the future mainly for us is to upscale for clinical trials and also globalize implementations, new technologies, data release, analysis software. So there's a lot of things going on with the flow cytometry. I think it's a good time to be in the flow cytometry service as well.